name's Adrian Dubuc. I'm the Vice Chair for Clinical Research in Cytogenetics and Genomics and the Director of the Clinical Cytogenetics Lab at Roswell Park Comprehensive Cancer Center. And it's my pleasure to have the opportunity to describe how we're attempting to raise the bar in lymphoma diagnostics through the comprehensive use of cytogenetic profiling. I'm sure this audience is well aware of what lymphoma is, but as way of brief overview, lymphomas represent a heterogeneous collection of malignancies resulting from the proliferation of lymphocytes. And these entities range from indolent diseases to highly aggressive variants with a rapid disease course. Now, diagnosis of a lymphoma is a complex topic and requires not only histomorphology, but the use of a number of ancillary tests, which can be both complicated and time-consuming. And ultimately, the challenges in lymphoma diagnosis, in part predicated on the number of entities that exists, can result in a misdiagnosis in up to 1 out of every 5 cases in the absence of expert review. And this, to me, highlights the critical need to improve diagnostics and to support the appropriate diagnosis such that every patient leaves the clinic with an accurate diagnosis. Now, cytogenetics plays a critical role in the diagnosis of lymphoma, and here I'm showing examples of the most recent iterations of the ICC and the WHO classification systems. On the left-hand side, I'm demonstrating with the red arrows how a number of different number of lymphoma entities now require the use, either detection or exclusion, of some structural rearrangements in order to achieve a final diagnosis. And the same is true for the WHO. In fact, morphology alone in some entities is no longer sufficient in order to achieve final diagnosis. And in the example on the right, it's the combination of morphology coupled with the presence or absence of BCL2, BCL6, or mixed rearrangement in order to achieve some of the final diagnosis, integrated diagnosis that are described. Now, what I hope to convince you over the course of the next 12.5 minutes or so is that the current diagnostic standards are insufficient. Now, in order to review those in brief, I think it's important to reiterate what our current options are. And fluorescence in situ hybridization FISH represents the current gold standard in the field. While this has been a well-used and battle-tested assay, it's important to note that it does have significant limitations that comes in the form of the need to have a priori knowledge of the specific targets that we're looking for. It also requires extensive validation of FISH probes, and this suddenly enters a different challenge and complexity in regards to the operational structure of a lab and balancing the time versus effort versus resources and volume effort to say, is this a test that we can afford to bring in-house? And unfortunately, FISH also suffers from both false negative and false positive occurrences, some of which I'll be describing over the course of my presentation. Now, while FISH primarily represents sort of the gold standard in the field, chromosome studies are occasionally used depending on the variant, sometimes in support of FISH-based studies. And again, when effective, it's incredible how much we've been able to learn in looking at the structure and the nature of the genome through this modality of testing. But like all tests, its limitations can be significant. And for chromosome studies, the requirement for cells dividing in culture, the resolution of the assay, and again, the propensity for false negative and positives are significant challenges to our ability to integrate this into effective diagnosis. Now, there's been a lot of excitement to what genomics and transcriptomics could do to the field of cancer diagnostics and cancer treatment. And while there's been incredible promises, particularly for myeloid malignancies, we haven't seen the same benefit occur in the context of lymphomas. For DNA-based short-read strategies, unfortunately, the short-read nature sometimes precludes the accurate detection of structural variants or challenges it in the very least. Well, RNA-based strategies have been incredibly effective for detecting in-frame gene fusions, but can struggle with enhancer hijackers, where we simply have overexpression. And enhancer hijackers is, again, the more common of the structural variants that we observe in lymphomas. All this to say that improved diagnostics is urgently needed for this disease type. Now, before I go on to share with you some of my experiences, my team's experiences with you with lymphoma, I did want to give a quick plug for some of my team's work that's being presented in the context of the symposium. And if you haven't seen Dr. Scott Ryle's presentation, I highly encourage you to revisit this talk, as well as the posters from Sam Brody, both you colleagues of mine who have worked in my research group and collectively have demonstrated, I think, the applications potential of OGM for a number of different hematologic malignancies. But moving back to lymphomas, I'd like to describe how some experiences from my past and my time in Boston is informing so much of my future in regards to our prospective work at Roswell Park. So we had a patient during my time in Boston, which came to us as a B-cell neoplasm. It really had a very sort of required a more robust workup and a better understanding of the specific type of lymphoma this was believed to be. But what both challenged and puzzled our hematopathologist was the presence of a DUSP22-ERF4 rearrangement that was described by outside FISH studies. Now, unfortunately, in our hands in Boston, I think that the DUSP22-ERF locus was not a probe that we had in-house, and so we weren't able to repeat those studies, and therefore karyotype ended up being our next best start to understand the true nature of this disease. I think it's safe to say you don't need to be a cytogeneticist to appreciate the complexity both in the string line, the ISCN that describes the cytogenetic findings of this patient, as well as just looking at the degree of variation that exists. But unfortunately, this particular profile is neither specific nor diagnostic. And now we're left with a challenge, which is to say that DUSP22 is typically associated with T-cell malignancies. We have this particular pattern, which is nonspecific. How do we reconcile the FISH results with the karyotype findings we have in-house? In order to answer this question, it's important to have some background in the understanding of this particular probe and genomic locus. So both the DUSP22 and ERF4 represent two genes that are located on the 6p25.3 cytoband and are separated by only 40,000 base pairs. Now, rearrangements of distal regions of chromosomes are notorious for being cytogenetically cryptic, that is, cryptic or invisible by GTG banding analysis. And thus, perhaps it was unsurprising that we didn't see any evidence of any alterations at that 6p ter band. But again, we're left with how do we reconcile the FISH results with the karyotype? We turned to an old friend in our particular cases, which was optical genome mapping, which has consistently provided us with a new perspective on cytogenomics. And on the left-hand side, I'm showing the matching karyotype for the Circos plot, on the right-hand side for the same patient. And a few points to me immediately jumped out. One is the degree of additional structural variants that exist and that are readily detectable through optical genome OGM analysis. But also that when we start appreciating the copy number changes, that many of the chromosomes we believe to be lost based on our chromosome analysis are in fact not lost, but just different. They have undergone complex rearrangements. And this ability to map and mirror both structural variants and copy number findings and bring them together ends up providing an additional layer of information, which is exquisitely useful in a number of the cases. Now, I like to think of OGM as a shotgun approach with surgical precision. And here's an example of exactly why this is the case. So in highlighting in blue, again, the same Circos plot for this patient, a particular rearrangement. This is an alteration between chromosomes 6 and 14. And what you can appreciate is that this results in, at the gene level, a rearrangement between the ERF4 locus and the IGH. So a cytogenetically cryptic translocation 614. And now, bringing this together, we can say, well, was FISH really false positive? How was it similar or dissimilar to those results? I think in order to fully understand this story, I think we have to appreciate maybe the anatomy of a FISH report. That DUSP22 and ERF4 are two genes that live in close proximity, but whose rearrangements are typically associated with different disease types. DUSP22 with T-cell malignancies, ERF4 is much more common in its rearrangement through B-cell malignancies. But the fish studies that we use are using probes that span nearly a 500 to 1 megabase region of the genome. And therefore, we don't have the clarity to be able to definitively see which of these genes within this locus is in fact rearranged. So while it's not uncommon that on a report for a gene like this or a locus like this, you'll see an annotation that suggests that DUSP22 ERF4 rearrangement was detected, perhaps more appropriately, this should be described as rearrangement of DUSP22 or the ERF4 locus. And I show this just to highlight that the current tools we use for lymphoma diagnostics, FISH in this case, can lack the specificity needed to confirm the rearrangements that end up being really critical. Now, the use of FISH and these false tests, I think, should be alarming for any of us who sign out cases clinically, and particularly for those of us who touch B-cell lymphomas and diffuse large B-cell lymphomas. Perhaps the oversimplified schematic of how this works for these types of high-grade lymphomas is when histology suggests a high-grade lymphoma, a battery of FISH tests, often MYC with BCL2 and BCL6, is performed in order to achieve a final integrated diagnosis. Now, what has been well established is that MYC break-apart FISH studies, which represents a critical mediator of aggressive phenotypes of lymphomas, can result in up to a 5% false negative rate. And I want to repeat this, I think, to let this sink in. I think because for our community, this is incredibly significant. MYC break-apart FISH studies can result in up to a 5% false negative rate. And this suggests that a number of lymphomas could be receiving an inappropriate or a misdiagnosis, in part because of the types of tests we're using. Above an example of exactly this instance that we observed during my time in Boston. On the left-hand side, I'm showing both a cytogenetically normal chromosome 8 as well as metaphase and interphase FISH that was unable to demonstrate any evidence of a MYC rearrangement. And yet, when we profiled this same tumor with optical genome mapping, we were able to resolve a small insertion of IGH into the MYC locus in such a way that it drove enhancer hijacking and MYC overexpression. So we began feeling more and more confident that optical genome mapping was giving us exactly what we needed, which was to say both the timeline and the resolution in order to detect the clinically relevant variants that exist in lymphomas. And because of this, in collaboration with Dr. Giorgio Inghirami at Weill Cornell and my previous chair, Dr. John Astor at Brigham and Women's, we underwent a pilot experiment in which we took primary lymphomas as well as matching patient-derived xenograft lines and said, how effective is OGM on solid tissues? I'm delighted to say that our ability to isolate high-quality DNA and subsequently label them was highly effective in PDX cell lines, and more importantly, resulted in a structural landscape which was expected for these TP53 deficient tumors, which is to say incredibly complex and chaotic. Equally important, in some of the PDX lines, we're able to identify pathognomonic driver alterations, in this particular case, an NPN1 ALK alteration. And I think what I appreciate about this technology is the ability to not only identify these clinically relevant driver alterations, but also on the left-hand side in the circles plots, identify the mechanism through which that occurs. In this particular case, a three-way rearrangement leading to this particular event. Now, I'm happy to see that as a promising preliminary results, we were able to demonstrate that for primary tumors, we were able to also isolate high-quality DNA, although I will say initially we were challenged with our ability to label these effectively. When labeling was corrected, what we were able to demonstrate was that primary tumors, that PDX lines, I should say, faithfully recapitulated primary tumors in terms of both the structural and copy number landscape we were able to achieve. And most importantly, primary tumors demonstrate we were able to detect the driver alterations that, again, ends up having so much diagnostic importance. And we expanded this series through collaboration with Sam Ng, who at the time was the Dana-Farber Cancer Institute and now at the NCI. And demonstrated that over a course of an expanded number of T-cell lymphomas, we could consistently and effectively, with 100% sensitivity, achieve and detect the driver alterations and also the type and nature of the rearrangements that occurred. To end my talk, I'd like to summarize that, in my opinion, lymphoma diagnostics have well-described limitations, but these are really hampering our ability to achieve appropriate diagnosis. That optical genome mapping has both the resolution and genome-wide coverage that far surpasses the technology that we're using, and that, in my opinion, OGM has the potential to be a first-line test for lymphoma diagnostics. So with that, I'd like to acknowledge many of my collaborators from my former institute in Boston, as well as my present institute at Roswell Park, and the collaborators who contributed to the study that I presented. Thanks so much for your time.